Yes, myself, Dr. Bharat Singh, working as the entomologist at Krishi Vigyan Kendra, Gurugram. As an entomologist, come subject matter specialist plant infection.
लाइन बता रहा था.
Okay, thank you very much. As the name of this important international conference, Advances in Agriculture and Biological Diversity Conservation for Sustainable Development. Integrated pest management play a very important role to maintain ecological balance and sustainable agriculture. Still, the farmers using not label claim pesticides in a disrupting manner that causes serious diseases and health hazards. Is the right way to all of us and environment. My big investigations, so based on the cauliflower, the cauliflower belongs to Prosperi family. It's an important vegetable crop of the district Gurugram, Haryana. Besides other constants, major constants are the insect pests which are responsible for crop losses. The farmers are using pesticides indiscriminately, resulting many problems like pest resistance, and secondary pest outbreak. We need to implement of IPM techniques by involving the eco-friendly management practices of the insect pest, cauliflower like diamondback moth and tobacco caterpillar. Survey of the crop areas with identification of major insect pests of cauliflower in district Gurugram. Usage of eco-friendly pesticides like botanicals and biopesticides. Implementation of the integrated pest management approaches against the target insect pest of the crop. The pests such as diamondback moth, tobacco caterpillar, cabbage semi-roofer, cabbage aphid, mustard aphid, painted bug, and are the important pests of this crop. Among the diamondback moth and tobacco caterpillar are the two major pests of the crop identified in the cauliflower throughout of the district Gurugram. Field survey in Gurugram district of Haryana for identification of the pests responsible for economic crop losses. There are four major blocks in the district: Pataudi, Faruknagar, Shona, and Gurugram. Layout of the experiment: insect pest complex associated with the crop already discussed. The major pests highlighted at serial number 4 and 5. Major identification of the identified insect pests like diamondback moth, damage symptoms in first slide and fourth slide, Dispodoptera vidura, mature larvae, observations of the different IPM modules at experimental field, interaction with the farmers about IPM modules and technological interventions. Yes, next. Dispodoptera vidura larvae feed on the cauliflower leaves and gregarious feeder in the nature. The infestation of this pest starts from the nursery itself. So this is the important. I need to take preventive measures at nursery stage to avoid the greenfield infestation. Mature larvae completely devour the leaves and bore into the heads. The moths of this insect gray in color, having the four wings with white strip lines. These are the larvae and damage symptoms of the two pests. These are also photographs of these Litura and Litura alostera insect. the damage symptoms on the leaves, observed percentage damage on plants, 30 to 40% in case of the Sporoptera, and in case of Sporoptera also 30 to 40%. Sorry. Extent damage of the another pest, Flutella gynostella, 30 to 50% on leaf damage. These are the IPO modules. 6 modules we have selected. To release of the under IPO module number 1, 2 modules of the Trichogramma palonis, that is the larval parasitoid of the pest. Second module is spray of the indosartan with the NSK neem seed kernel extract. IPM module number 3, 2 release of the Trichogramma palonis at the rate of 1 lakh per hectare at the interval of 10 days, and spray of the BTK, basis thuriges bacteria pesticide. Under IPM module number 4, spray of spinosad, 45% SC. IPM module number 5, spray of the cylohalothrin pesticides, and second is spray of Nima gel. Nima gel also the botanical pesticide, and in case of IPM module number 6, spray of the spinosad with the spray of Nima gel, and module number 7, untreated control. This is the efficacy of the different IPO modules. After first and second spray, after third and fifth and 7 days, before spraying of the before application of the IPM modules, damage symptoms of the pest 8.5 7.67 7.83 larvae per average larvae per 5 plants, and in the last average reduction of the after first and second spray reduction of the larvae 3.66 1.41 2.88 0.16 and 1.91 0.61 7.61 in case of untreated control. This is the same application of the IPM modules in case of the Sporoptera. This is the economics of the IPM modules of the cauliflower third yield. Highest net profit obtained in case of IPM module number 4, that is 38,325 rupees per hectare. Conclusion: in present investigation, it was observed that tobacco caterpillar and Sporoptera litula and the diamond back moth were the major pests of the cauliflower throughout the 4 district of the Gurugram, and damage observed 30 to 50% in case of the Flutella gynostella, and in case of Sporoptera litula, that is 30 to 40%. And IPM module number 4, comprising of the spray of this spinosad and NSK at the rate of 5 ml per liter water 10 days after first spray, and again repeated spray of NSK after 10 days, highest gross income observed. Thank you very much.
Thank you, Bharat Singh, for your nice presentation. Then we'll move on to our oral presentations. So, Nidhishri is here. Nidhishri, then after that, Bharati. Bharati. Okay, be ready. Then Nagaraja. Okay. Mahesh Kumar, Mohan Kumar. Okay. So Shrija. Visa is valid. Okay. Yeah, start. We'll give 3 minutes for 1 minute for discussion. Totally 4 minutes.
Good afternoon, dignitaries. I'm Dr. Nidhishri and I'm a doctor of yoga and naturopathy. And my topic of presentation is how yoga and naturopathy, the Indian system of medicines, contribute to sustainable development, holistic approach for environmental and human health. So it is an interdisciplinary approach. Naturopathy is an Indian system of medicine that believes in the inherent potential of the body to heal itself provided the favorable conditions are given. So it is based on the philosophical construct of Pancha Mahabhutas. The principles are that the healing power of nature, for example, sunlight is a good source of free vitamin D3. So, and moderate exposure of sunlight is shown to have beneficial effects because of the increase of vitamin D. And to treat the root cause of diseases and not just suppress the symptoms. Doctor as a teacher, so the word dossier is to teach, meaning the doctor teaches healthy habits, and prevention is better than cure. And food is the best medicine because gut health is the best health. So linking this to organic farming, that is very important for sustainable development. Various nutritional and health advantages are seen. Coming to nutritional advantages, organic crops are shown to have high antioxidant level like polyphenols, and also omega-3 fatty acids are high in organic dairy products. Coming to health aspects, organic intake reduces the incidence of infertility, preeclampsia, and other diseases. So, better to promote organic food, and also it has positive effect on gut microbiome, which has beneficial effect on the good bacteria, have beneficial effect on cognitive health, physical health, and emotional health. Beyond organic farming, the naturopathic institutions are an epitome for biodiversity conservation because they conserve medicinal plants and are into organic farming. Various case studies I have presented, like Jindal Institute of Naturopathy in Bengaluru has 100 acres of organic farming which they use for their patients. And also SDM Naturopathy Institute in Uchre, Ayurved Hospital of JSS in Mysore, they have around 300 species of medicinal plants. So this is one of the example of biodiversity conservation. And yoga, the mind-body medicine, the philosophical construct sourced from the Ashtanga Yoga gives foundational principles for biodiversity conservation, sustainable development. Like the Ashtanga Yoga, yamas and niyamas, ahimsa and shaucha, that is the niyamas, aparigraha. Ahimsa not just for against human conflict, but human-animal conflict against poaching, habitat destruction, and even erratic deforestation. And the aparigraha, non-possessiveness, that is against illegal encroachment. Shaucha, not just human cleanliness, but environmental cleanliness, that is solid waste management, to beat plastic pollution. And also pranayama needs toxic free air and atmosphere. So this is the interdisciplinary or interdependency between human health and environmental health. And how naturopathy and yoga contributes to sustainable development. SDG goals 3 especially, to promote health for all, the non-communicable diseases under SDG 3.4. Yoga and naturopathy has various researchers to prove that the modalities have beneficial effect to hypertension, diabetes, other autoimmune diseases. For example, naturopathic modalities like clinical nutrition, diet therapy, fasting therapy, acupuncture, herbal medicines, physiotherapy, massage therapy, hydrotherapy, these are undergoing various rigorous research to show their benefits to human health. And coming to SDG 11, sustainable cities and communities, like green lifestyle, promoting home gardens, public parks, and Ayushman Bharat Jan Arogya Yojana has promoted health and wellness centers which are promoting preventive and curative therapies like yoga. And coming to climate action, so SDG 13, promoting organic foods, regional and seasonal foods, and green lifestyles, nature-based solutions, reduces carbon footprint. Various advances in naturopathy and yoga in the form of researchers in CCRY, Central Council for Research in Yoga and Naturopathy, and integrative medicine departments in AIMS, New Delhi, and NIMHANS, and National Medicinal Plant Board under Ministry of Ayush is in collaboration with MOEFCC is conserving a lot of medicinal plants to conserve for better biodiversity conservation. So, in conclusion, I want to say that the need is for action. That is, prevention is better than cure. It is a medical prescription, and long-term solution is nature-based solution. So, sustainability comes through successful implementation of nature-based lifestyle and therapeutic approaches. Thank you so much.
Thank you, Pooja, for your nice presentation. And you're affiliated to which university?
So I'm a naturopathy and yoga doctor. I worked as a senior research fellow in Central Council for Research in Yoga and Naturopathy. So I'm basically a researcher, and currently I'm an integrated medicine physician.
Presently, what you are doing?
Integrated medicine physician in Ambara Wellness Clinics. I'm interested in interdisciplinary researches. I've done my master's in psychology, so I've connected naturopathy and yoga, the system of medicine, the Indian system of medicine, how it contributes towards sustainable development and biodiversity conservation.
Good. Because in the present days, it's very important that health is the most priority to maintain a good health rather than going to doctor, taking medicines, drugs, and all. So the best way, natural health maintenance or healing, that is through yoga and naturopathy. So well known. Our ancient Hindu times, all Vedic. If you go literature, it has come from India. Yoga, natural, but now it is spread worldwide. All these advanced developed countries, Europe and America, they are now following, and they have become experts. Okay, thank you for your
Thank you
presentation.
so much for the opportunity, sir. Thank you.
Yeah, next person, Ms. Gaikwad, I think Miss, right? Yeah. Gaikwad Priyanka Anil. Yeah, number 5.
Good afternoon, everyone. I am Dr. Priyanka Eka. I am from Maharashtra. Topic of my presentation is features of Tribulus terrestris medicinal plant and their allopathic effects on seed germination and seedling growth of maize and wheat. Introduction. The term allopathy, which means allelon meaning one other and pathos meaning who suffer, referring chemical inhibition of one species by another. Then material methods. For whole plant leachets, I firstly collect plant, then washed with distilled water, and leachets were prepared. Then statistical analysis were done. Then LCMS analysis were carried out for the identified phytochemicals. Then this is a photograph of my plant. Then the results and discussion, in which table 1 exhibit effect of leachets of Tribulus terrestris on seed germination, and second exhibits the effect of leachets of Tribulus terrestris on seedling growth of maize and wheat, in which we can observe as the concentration goes increases. Concentration goes increases, the effect of leachets is going to be increases, and seedling growth as well as seed germination percentage is decreases accordingly. Then statistical analysis. Then these are the photographs of my research, in which at control in the control plate we can see germination percentage as well as seedling growth is maximum, and as the concentration going to increases, percentage of seedling growth and seed germination is decreases. Then allochemicals reported from Tribulus terrestris whole plant by LCMS analysis, in which we can observe fourth number compound, that is fumaric acid, which is their IPSC name. This compound is abundant and its retention time is 3.65 and shows highest MX spectra. Then LCMS speaks of whole plant extract. The summary and conclusion. So, summary, this plant is collected from the Baramati Tehsil in leeches. Say the leeches of Tribulus whole plant had an overall suppressive impact on the seed germination and seedling development of both maize and wheat. Higher concentration of these leeches significantly reduced seed germination rate. The most pronounced inhibition was observed in with Tribulus terrestris leeches at 15% and 20% concentration, particularly from the root, stem, leaf, and whole plant leeches. Wheat seedlings exhibited a greater reduction in seed germination percentage and root shoot length compared to the maize seedlings. These are the references. Thank you.
Thank you, Priyanka, for your nice presentation. Yeah, we'll move to next presentation by Bapu Kundik Ocha. Yeah.
Yes, ma'am. Yes. Okay. Good afternoon, all. Respected chairman and dignitaries of the diet and all my dear participants. I am Professor B.K. Avsar from Vidya Pratishthan College, Baramati, district Pune, which is affiliated to Savitribai Phule Pune University, Pune, Maharashtra. I am going to present my topic, the art of cultivating bonsai, a tool for biodiversity conservation. Bonsai is an art form that comes from ancient Asian culture, originating in China and developed by the Japanese. In the 13th century, the Japanese collected and potted wild trees that had been dwarfed by nature. That these naturally formed miniature were some of the first bonsai. A bonsai is a literal a tree in a pot, which is further imitates in miniature the appearance of an old tree in a nature. Old specimens in nature, unlike juvenile trees, have compact, rounded tops and horizontal or drooping branches, which makes them appear aged and graceful. These bonsai art refers to the cultivation of miniature trees, which may be trees or shrubs and vines, can be manipulated and trained to grow in a way that it mimics the nature form or mature plant. The pruning, fertilization, and are the two essential activities for the successful management of bonsai cultivation. Both shoots and roots of the plants must be regularly and constantly pruned to achieve the desired shape and control the plant's growth. Bonsai trees are generally cultivated for outdoor environments. Therefore, when used indoors, the necessary suitable environmental conditions must be provided to ensure their proper growth and development. This study was carried out to promote the bonsai making activities in the students and general public by cultivating ornamental plants, fruit plants, and endemic tree species. Bonsai enthusiasts creates living repository for genetic material. This form of ex situ conservation acts as a safeguard against the loss of species in their natural habitats. Bonsai serves as an educational tool that promotes a deeper understanding of plant biology, ecology, and biodiversity. The practice fosters the appreciation for the nature's delicate balance and encourage conservation action. The long-term care required for bonsai cultivates patience, long-term thinking, and a sense of responsibility for the living things. This mindset can translate into broader environmental awareness and engagement with the natural world. Uh, for the material method, we have collected many plant species from around Pune district or from Baramati district. These plants were collected in the form of stem cuttings or grafted seedlings, naturally occurring mature forms as well as nursery saplings. And these plant specimens were treated for making different styles of bonsai by applying standard procedure using various types of bonsai pots. The bonsai soil is prepared by using garden soil, brick pieces, sand, charcoal, chalk pieces, and cocoa peat in different proportion for different plant species. The mixture used is fast drainage and moisture retention substrate that provides excellent aeration for the plant roots. Unlike unconventional potting soil, it is designed to mimic the well drainage mineral soil found in nature, which is essential for bonsai confined to small pots. Organic matter like vermicompost or farmyard manure, peat moss, and coconut coir are added sparingly to increase water retention. Inert particles like crushed granite and are also added in the pots. These tools were used for uh, taking care of the bonsai. The bonsai techniques, there are two types of technique. Collecting the bonsai, bonsai may be collected from the nature, or which is called as bonsai hunting, and bonsai can be prepared by seedlings or cuttings or layerings or grafting from, which can be brought from nurseries. Uh, there are these are the certain procedure for while maintaining the bonsai, that is pruning, wiring, repotting, leaf miniatures, watering, feeding, pinching, plant protection as well. Our results and discussions are there are eco-psychological practice regarding the bonsai preparation. Research include that the participating in the bonsai art has positive impact on an individual's well-being, suggesting it as an therapeutic and healing practice with minimal resource requirement. And it has also aesthetic values or aesthetic goals to main. The main objective of bonsai aesthetic is to create miniature that appears old and naturally weathered. Bonsai can be classified into about 10 types that we will see. These are some types, like formal types of bonsai, informal type style, slanting style, then cascade style, semi-cascade style, this broom style, then wind swept style, literati, twin trunk style, and forest styles. Plant selection, it is a safe to begin the common plants that we do will in your area. Be sure that the plants you consider meet the requirements for good bonsai. Some old favorite plant species, which are junipers or ficus species, bougainvilleas, boxwood, desmodium, rain tree. There are many species which are terminalias, Delonix, Lagerstromia, etc. So this is bonsai hunting. The pots of different sizes or shapes can be used from different materials. These we have prepared around 170 bonsai of 62 species. These are the some botanical names, their family, and the vernacular name and number of individuals we prepared the bonsai. These are the some photographs of bonsai we prepared in our college or in garden. These are the bougainvillea, ficus religiosa. These are the some bonsai which are prepared by me and my students. This is Lagerstromia speciosa. We call it as a taman. This is a state flower of Maharashtra. It is found in the form of very large tree, but we prepared in the form of bonsai. Then these are this is ficus rampai, which is called as bodhiruksha. These are the initial stages, and this is the final stage. These are some tecomas. This is Syzygium family of jamu. These are also some different colors of bougainvilleas. This is desmodium, again bougainville. This is terminalia mentally. It is an ornamental plant which is cultivated around in the as a roadside tree. This is lantana camera, which is found in this area as a wild plant or as a weed. This is Lagerstromia. This is Muraya paniculata. It is a flowering plant, a sweet scented flowering plant. This is Ficus benjamina. This is Ficus revoluta. These bonsai, their age is around 8 to 10 years. This is hibiscus rosa sinensis. This is a common hibiscus. This is Ficus microcarpa. I have taken this cutting. This bonsai is prepared from stem cutting, and the mother of the this plant is around 200 years. These are some Tamarindus indica, Ficus microcarpa, Carissa corundus. This is Ficus religiosa. This is Mangifera indica. It is around 5 to 6 years and it shows fruiting. This is Citrus lemento. This is Delonix regia. These are the again some bougainvilleas. This is bougainvillea. This is again this is Terminalia mentally. This is Ficus audumber, Ficus racemosa. This is people. This is Ficus bengalensis or banyan tree. Then this is Adansomia digitata. It is very large trunk tree. I have prepared this bonsai. Then these are the certain. This is Samania saman. This that is Arjuna, terminally Arjuna. Both these plants are terminal Arjuna. This is a forest type of bonsai. So these bonsai can be used as a ornamental plant in offices or in corridors of your college or hotels. And lastly, bonsai, the art of cultivating miniature trees, serves as not only as a aesthetic for practice but also as a tool for biodiversity conservation and environmental education. By propagating rare and endemic species, bonsai collection function as a living gene bank that helps preserve plant diversity and protect specimens from extinction. Ethical sourcing and the use of native plants enhances the ecological benefits. Originating in China and Japan, bonsai involves training and maintaining young trees through pruning, root pruning, and specialized care to care create mature natural forms that can live for centuries. In a study involving 169 plants from 62 genera, bonsai cultivation was promoted as a hands-on activity to raise environmental awareness. Beyond conservation, bonsai fosters understanding of plant biology and ecology by teaching patience, responsibility, and sustainability, ultimately strengthening human connection with nature and supporting environmental stewardship. Once Chinese scientists rightly said that don't make the tree a bonsai, make the bonsai a tree. Thank you so much.
Okay, thank you, Babu, for your good presentation.
Thank you so much.
Any questions from the audience? Okay, okay, thank you.
Thank you so much.
We move on to next presentation. Nine, no, serial number. Okay. Bharati, please. So you will get 3 minutes plus 1 minute finally to conclude. Okay.
Good afternoon to anyone present here. I am going to present my research titled Evaluation of Marine Endophytic Glycomyces Species for Sustainable Crop Growth and Biocontrol Application. And I'm skipping this introduction section. The background of the study is that endophytic actinomyces has been exploited for many biomedical application, but for agriculture purpose, it has not exploited. So I chose sea grades and seaweed associated endophytic actinomyces for sustainable agriculture. And my objectives includes to isolate and characterize endophytic actinomyces and to evaluate plant growth promoting triads and study the seed germination and pot experiment to assess the antimicrobial, antibiofilm, and anti-insecticidal potential of the selected endophytes and to perform GCMS analysis for their profiling the bioactive secondary metabolites and the identification of potential strain by molecular techniques. And I'm skipping this methodology and directly going to results. I will explain the methodology itself. This is the sampling site. I collected the seaweeds and seagrass from Gulf of Mannar. And these are the seagrass and seaweeds that I have collected. And the distribution of the actinomyces shows that streptomyces is found to be dominant, followed by monospora. And this is the plant promoting traits of the actinomyces. Among the 21 isolates, the SGT7 showed maximum PGP activity with high IEA activity. IEA activity was tested by using L-tryptophan growth and phosphate solubilization by Picasso together, and nitrogen fixation by nitrogen-free Jensen medium was used, and ammonia production was assessed by using peptone water. And it shows the SG7 showed maximum activity followed by SGR6 and SGT3. And this is the antibacterial activity of endophytic actinomyces. I have chosen Rostrania solanicerum and Pseudomonas cichori, Xanthomonas citri, and Pseudomonas syringae for assessing the antibacterial activity. Among that, the SGT7 shows maximum inhibition against all the distinct pathogens, and the MIC MPC also revealed the same. The lowest activity was regarded against this Xanthomonas citri and Pseudomonas syringae. And this is the anti-biofilm property of the actinomyces, and biofilm inhibition assay was performed by using crystal valley technique, and the protein leakage was performed by using Barford technique, and the APS was quantified by using ammonium sulfate method. And among this, I found the dose-dependent biofilm inhibition was performed, and the maximum inhibition was recorded against Rostrania solanicerum and Pseudomonas cichori. And protein leakage assay also revealed the same at the concentration of 150 microgram per ml. It shows protein leakage of 95% after 20 hours of exposure, and EPS quantitation show there is a significant reduction in EPS formation.
And these symptoms indicate that glycomyces species inhibit biofilm formation by damaging cell membrane and suppressing the EPS synthesis. And this is a seed germination assay. I used Vigna Munda and Elianthus anus for my study. And it shows that dose dependent activity at the maximum of 0.5% biomass, the seed germination showed remarkable. And the optimum inoculum level was optimized at 0.5%. After that, I couldn't get any enhanced results. So I optimized the inoculum level to be 0.5 percentage. And these are all the experiments that I have conducted, photographs. And the insecticidal activity, I used Bimisia tabaki since it infects most of the plant. And it shows that the high mortality was observed after comparatively the positive control, and lower concentration has minimum mortality of 20%. And this result confirmed the anti-infective potential of the glycomyces metabolites. And this is a GCMS analysis. I have obtained 10 different compounds of those benzaldehyde and the pyrimidine derivatives shown to possess antimicrobial, antibiofilm activity, and we are in the process of docking this thing. We are going to use this chemicals as ligands against insect protein and bacterial protein. And this is the potential actinomycete site isolation, and this actinomycete that is present in ISP2 agar. And molecular identification reveals that it belongs to glycomycete species. And to the conclusion, endophytic actinomycetes from sea trees and seagrass exhibit maximum PGP and biocontrol activity, and among them glycomyces shows potential activity, and it was the first report of marine glycomyces possessing this type of PGP activity, and it shows high IAA efficient phosphate solubilization, nitrogen fixation, and ammonia production. And crude extract also showed to process antibacterial, antibiofilm activity, and it has showed insecticidal activity against benzodiazepine, and the GCMS profiling showed the benzaldehyde and pyrimidine derivative, which was a potential targets for against pest and bacteria phytopathogens. I conclude that glycomyces should be possibly applied as a promising biofertilizer as a biocontrol agent for sustainable agriculture.
Okay, thank you. Any questions from the audience? Okay, if there are no questions, thank you once again.
Thank you.
And so we will move on to next presentation by serial number 10, Nagaraja O. Yeah.
Good evening. So my name is Dr. Nagaraj O, Assistant Professor, DRM Science College, Davangere. So detection, location, and seed transmission of Macrophobia facialis in Niger seeds. So Niger is one of the most important bird seed. It belongs to the family Asteraceae. So this is the seeds from extracted from useful for chutneys, sambars, and etc. etc. So materials method. How to detect the fungus Macrophobia faciolina? So there will be detection of seed-borne Macrophobia faciolina by seed egg test methods, standard bladder method, potato dextrose method, water agar method, and then 2,4-D methods. And then location of the fungus, the Macrophobia faciolina by component plating methods. The component platings are seed coat, cotyledons, and plumule and radicals. These component plating method, we isolate the causal agent of Macrophobia faciolina fungus. Seed transmission of the fungus. So this methodology is studied in experimental plots. So this is transmission studies in the experimental plots. This is the incidence of Macrophobia facial and other fungi in seed egg test methods in Niger seeds. So seed component plating methods. The Macrophobia faciolina is higher incidence of seed coat and then cotyledons only. And then plumule and radicle, and plumule and embryonic axis. So more number of this fungus is adopted in fertilizers and seed coats. This is transmission. So this is transmission study in comparison between 2 years, 24 and 23. So this the seed transmission, the 2024 is more disease transmission compared to the during 2023. So this is the conclusions. Detection of seed borne Fagellina and other microbes play an important role in determining the quality and longevity of seeds. Microbial invasion can lead to rotting, loss of seed viability, germination, quality, productivity, and yield. It suggests that seeds are major agent of fungal transmission. Seed should be treated with suitable chemical before sowing to reduce the fungal infection. This is due to the environmental factors like rainfall, humidity, temperature, pH, and also growth stages of the crop. Seed pathology involves the study of living entities, environmental factor affecting the adversely to the seed production and utilization, as well as disease management practices applied to seeds. Finally, Macrophomia fagellina, the causal agent of root rot disease of Niger crop. This is my horse.
This study you have done, Niger.
Niger.
Yellow.
Puccedo ಅಂತಾರೆ sir. Puccedo. Visocia abyssinica. Visocia, this is a plant. Visocia, transmission studies in the plants. And then seeds.
Okay. Thank you. Next, Mohan Kumar. You are Mohan Kumar.
Yes, sir.
Respected Chair and dear delegates, on a special occasion, I'm going to present my work on evaluation of antioxidant properties of fruit bodies and mycelial extracts of Coprinopsis cinerea, an edible coprophilous mushroom. I would like to thank Professor Savita Janaki Raman, former head and chairman of Department of Microbiology, Bangalore University, for her guidance. So all of us know that the antioxidant plays a crucial role in so most of the food applications. Whereas we require an antioxidant which is having which is drawn from a particular a safe manner. So in that juncture, so we need an antioxidant. So that should be obtained from some of the important natural resources. So one such resources, what I have used here is, so extraction of a mushroom from the coprophilous, that is coprophilous mushroom, the Coprinopsis cinerea. So that I have extracted. So in fact, this mushroom grows in the cow dung. So then these are my objectives of the study, where I isolated Coprinopsis cinerea in the buffalo dung, and I have cultivated this Coprinopsis cinerea strain in a wheat flour medium. So this is one such medium. So we have developed in the laboratory, and we have extracted Coprinopsis cinerea using methanol, and also performed antioxidant properties. And these are methodologies followed for isolation of Coprinopsis cinerea from the host, from the buffalo dung. We have cultivated using specially designed media developed by us, wheat flour broth media. And we extracted metabolites from methanol, and also evaluated antioxidant and its free radical scavenging, total antioxidant capacity, reducing power. And we also characterize the antioxidant components using SPLC as well as GCMS. To the results and discussion, we have come across several species, and in that we have C3 Coprinopsis cinerea, we have submitted our result into the GenBank Open Accession number. And this is something which we have cultivated in the laboratory, where you can see the growth of Coprinopsis cinerea even in the Petri plate. And we have obtained the Coprinopsis cinerea mycelial biomass. In fact, at the expense of 50 paise, we could able to get around 80 grams of mycelial biomass. That is something which is an innovation in our research. And we extracted the extracts using methanol. And we conducted the radical scavenging activity using DPPH and ABTS, as well as hydroxyl radical scavenging activity. Also, we have used different types of the standard antioxidant in order to compare the antioxidant properties. These are the results of the antioxidant properties, whereas we have found that the fruit body is having 5.39 mg per ml of antioxidant, that is DPPH scavenging, compared to all other extract. Even mycelial extract also has shown, little bit more. And whereas ascorbic acid, BHA, BHT is it is very good antioxidant, but whereas it's a crude extract has shown a good antioxidant property. And this is reducing power using methanolic extract of Coprinopsis cinerea. It also showed a very good reducing power properties. And we also went with a total antioxidant capacity, whereas in that the fruit body has shown around 2.04 mg ascorbic acid per gram of dry weight compared to mycelial extract. And the total phenolic content and flavonoid content also found to be very effective in case of fruit bodies. It has shown around 2.08 mg per gram of dry weight in gallic acid equivalent. And the total flavonoids found to be 0.71. And we have correlated using the Pearson correlation efficients. We found C2F is correlated, the TPC is correlated with the DPPH and the ABTS activity based on Pearson coefficient. And we also found out the antioxidant properties of the Coprinopsis cinerea, whereas we found HPLC results presence of ascorbic acid, gallic acid, vanillin, vitamin A, and butylated hydroxy tolerance were detected using HPLC. And similarly, in case of the fruit body and mycelial extract as well. And we want to know some of the components which is present in GCMS. We went with GCMS analysis of fruit body extract as well as methanolic extract. We found presence of stearolic acid, around 34.52% of stearolic acid. And in fact, the resorcinol is found to be 16% in the extract. And similarly, for mycelial biomass, found to be three major component. So something which is 33% is succinic acid, and 14%, 13% is stearolic acid. To summarize, the Coprinopsis cinerea, this is the first report that the extraction of mycelial Coprinopsis extract used as an antioxidant. And also, we found out presence of 16 major chemical constituents. Overall, this can be used, the methanolic extract of Coprinopsis cinerea can be used for any other applications. So thank you very much. Any questions, clarifications?
Can you go to first slide? First slide.
Yes.
Let's see.
Yeah.
First title, please. No, okay.
Ma'am? Yes, it is edible. It is edible, something which is basically it is not considered to be edible because of the hygienic. But what did we do in our research? We have incorporated same thing that in the media. We have designed a media, it is wheat flour broth. So that particular it just has a wheat flour and some other micronutrient. So we wanted to bring out from that unhygienic to hygienic. Now it is exclusively growing. Like you can see even in our Petri plate. So and also easily you can grow it in mycelial and that can be extracted. So it is edible. In the East countries and all people are eating this particular dish.
At Saint Joseph's University, you have a department called Food Science and Technology.
Yes, sir. Yes, sir.
Since how many years the department is open?
This is a sixth year. 2019 we started. Sixth year.
So how you said that the phenolics and flavanoids. How we have analyzed, extracted procedure method.
So it is a malleolic extraction. There is a cold extractions method. So we've used a cold extraction method. Of course, oxalate extractions also we did, but cold extractions method is maceration. The initial extractions method that we did using methanol. So then that we went for analysis. So
Thank you.
thank you very much.
Next presentation. 12, serial number 12, Sija, UM. Come.
Good afternoon, all. I am Dr. Shija. I am from Kenya University. I'm a postdoctoral fellow. And here I am going to present my a small part of my research work. Is Bacillus valesensis as a potential endemopathogen of Spodoptera littoral. Okay. We all know that Spodoptera littoral is a littorious agricultural pest, because it was found to infest more than 120 economically important croplands. So managing these pests is very it is challenging because of its high survival rate, high fecundity, and its voracious feeding habit. And our dependence or reliance on pesticides, it will result led to soil degradation and development of pesticide resistance. So in here, the study aims to explore the biological control methods, specifically using endomopathogenic bacteria. My study is to study the endomopathogen. Objective is to study the endomopathogenicity of Bacillus valesensis on Spodoptera littoral in the laboratory condition. The materials and methods includes first method includes rearing of Spodoptera littoral in laboratory condition, and second was isolation of bacteria from soil. The Spodoptera littoral was reared in laboratory, carefully as food source, and for this rearing trays are prepared by using soils collected from 7 different sites such as paddy, household, mangrove forest, etc. And it is covered with a muslin cloth. Leaves were replaced daily to ensure freshness and hygiene, and larvae are monitored regularly. Once mature, the larvae burrow into the soil to pupate, and emerging adults are transferred to a mating cage with cotton swab soaked with a 10% fine solution for feeding. These are the different life stages of Spodoptera. The ECMAS. A single female can lay about 200 to 300 eggs per in her life. And these are the second instance of Spodoptera. And this is the fully grown larvae, this pupa, and this adult one. For isolating in laboratory, we have isolated 10 bacteria from different soil types, and it is labeled as B1 to BS1 to BS10. Each bacterial strain was introduced into sterilized soil in separate boxes, and is larvae of Spodoptera littoral and pupae were introduced into each box. Mortality was recorded daily for a period of 5 days. Larvae that died. Both bacteria were isolated from both the dead larvae and the corresponding soils via seeded dilution and streaking on nutrient other plates. These are the bacterial isolates from soil. This is the Bacillus valesensis. Observation and result. The most effective isolate was BS6, and it was selected for molecular identification and confirmed as Bacillus valesensis. These are the observations. Percentage of mortality. High percentage of mortality was observed in the BS2. BS6 and BS6 was selected due to high mortality across life stages, consistency of pathogenicity, and the larval mortality pattern. The infection caused by Bacillus thuringiensis exhibited a time-dependent pattern. No mortality was recorded during the first 2 days. On day 3, 3 larvae were found dead. That is indicate 12%, indicating the onset of infection. Mortality increased to 5 larvae by day 3, and cumulative death was observed 10, peaking at 40% by day 6 with a 10 cumulative deaths. No further deaths occurred on day 7 and day 8, suggesting the infection has stabilized and the remaining larvae may have direct resistance or were not susceptible to the bacterial isolates. This is the person mortality pre-pupal stage. Pre-pupal mortality was tracked over 3 days with an initial group of 15 individuals. No mortality was observed on day 1, and the one pre-pupal died on day 2 and 2 more day 3, resulting 20% total mortality. This suggests that Bacillus thuringiensis affects pre-pupal but with a slightly lower pathogenicity than in the larval stage, possibly due to physiological changes as the insect transitions between stage 1. This is the table showing the mortality rate in pre-pupal. Then pupal mortality. Significant mortality was observed in the first 3 days. No pupal mortality occurred after 3 days, indicating bacterial activity was largely limited to early stages of pupation. This is the table, and these are the photos showing the infected larva and pupa, and larva showed decay on the bacteria due to bacterial decay activity. This is the pupal stage infected by the bacteria. Conclusion. This study is relevant because it explores a sustainable biological method to control Spodoptera litura, a major agricultural pest. It supports integrated pest management by identifying Bacillus thuringiensis as an anti-endopathogenic bacterium. We know that Bacillus thuringiensis is a non-biocontrol agent which is used to control plant pathogens like bacteria and fungi. It is also a plant growth promoter because it promotes the production of auxins in plants. And it is also has cellulase and protease activities, and it can be used in many food industries and pollutant degradation. Also, it can also control as to control can use to control plant pathogens. So this observation it added the a plus point to the positive quality of Bacillus thuringiensis. That's all.
Thank you. Any questions from the audience? Yeah, please.
NPV is already potential pathogen provider, especially with respect to your product.
Sorry, ma'am.
What is the potential effectiveness of your strain against the bacteria compared to that of the SNNPV? Nuclear polyhydrosis virus, it's already established, and also the farmers are very much using in the field condition itself against this Spodoptera litura.
Because I take this topic as Spodoptera litura is taken as a lab specimen for my study. Actually, my purpose was to isolate endopathogenic bacteria from soil. So in this laboratory, I used Spodoptera litura as a experimental specimen.
Yes, ma'am. Yes, I'm not asking that one. the potential difference between your test organism, that's the
It
bacteria.
can be easily used, sir, because the Bacillus valencix is already present in the soil, and it can be cultured easily, and it can be.
No, my question is, can it be used as a surpass over the HMPV?
No, sorry, ma'am, I am not.
Okay, thank you.
Thank you, Teresa. Then Shiva Kumar, serial number 17, Shiva Kumar KM.
Good afternoon to Chairman, Co-Chairman, and their delegates. I would like to present my research paper on impacts of different strategic nutrient management practice on yield of Paraganati in Uttara Kannada districts. So all of you know that Uttara Kannada district is one of the important agricultural and horticultural crop growing district. It comes in located in western part of Karnataka, rich biodiversity, plant diversity, soil microorganism. Among these horticultural cropping system, it is the most important or most profitable cropping arecanut cropping system in the Uttara Kannada district. So this area usually generally characterized by high rainfall area, more for yearly per annum received more than 5,000 to 4,000 to 5,000 mm rainfall meter. Then altitude variation and temperature fluctuation, other factors. Then generally arecanut crops require larger quantities of nutrient to support its growth and yield of the. This arecanut generally have a high nutrient requirement crops. Then and it is predominantly grown in acidic lateritic soils. So these soils generally have an inherent soil constraint. For example, pH fixation, phosphorus fixation, leaching of basic cations, and low acidic pH. This leads to the instability of the nutrient retention and poor cation exchange capacity. Then further, higher removal of mineral nutrition of this cropping system that also leads to the instability of the crop nutrient imbalance in this system. And recently, in these reason, the increase evident to sustain the soil health for providing good nutrient management practice. For our Uttara Kannada district farmer, the maintenance of quality of health and the soil health and plant health mainly through adopting best nutrient management practices. So among the practices, generally the Uttara Kannada district farmer they are practicing for arecanut cultivation adopted integrated nutrient management practice, then organic farming, and also the recently farming natural farming nutrient methods are formed. So these nutrient management practices definitely will influencing on soil health as well as arecanut crop yield. So in this background, we conducted the research to assess the different nutrient management practice on the yield of arecanut yield in under acidic soils. So I have chosen for the study the Uttara Kannada district of Sirsi and Siddhapura taluks of Uttara Kannada district. I have identified initially I have identified 20 farmers for 3 nutrient management practice adopted by that is INM and organic farming and natural farming. Then after the initial soil survey, we collected soil sample from the surface soil sample that is 0 to 30 centimeter depth and then analyzed the nutrient status that is NPK, calcium magnesium, and also to assess the new yield of with respect to nutrient management practices. So coming to this, the after analyzing these are the through questionnaire I collected the information about how much of nutrients added through organic and inorganic and natural farming. With respect to INM Aracanite farmer, they are applied that is 240 to 454 kg per hectare for nitrogen, 142 for phosphorus, then 276 for potassium in respect to natural farming 150, 80, and 123 kg per hectare NPK are applied. Coming to the organic 230, 117, and 173 kg of nutrient added through organic and also fertilizers. So coming to the result and discussion, generally, INM farmers recorded highest the NPK content that's available, nitrogen available, phosphorus available, that is about 242, 36, 142. With respect to yield, if the nutrient management practice is very good, the arecanut yield is also increased. The greater arecanut yield is recorded in INM practices, that is 32 quintal. For organic farming, they recorded 92 quintal. Then ZBF farmer, there is a natural farming farmers are recorded only for 11 quintal per kg hectares are recorded. This is the graph show that available nutrient status with respect to areca yield. Generally, INM practices recorded higher yield and higher nutrient status. Then also with respect to same result, what trend was observed in phosphorus and potassium. So ultimate the summary of the conclusion of the ultimate aim of the any cropping is not only to obtain the additional income but also have to see the to maintain the good soil fertility for, status for long term. Though soil fertility is largely determined by largely by input usage of organic or inorganic. My suggestion is for any for with respect to yield oriented for to feed the growing population or to adopt organic, that is organic with fertilizer application. With respect to soil diversity, soil health, you practice slowly, practice organic farming and natural farming is the best. Thank you, sir.
Yeah, thank you, Shivkumar. Uh, any specific reason to select Uttara Kannada district for your study? Because you are working in horticulture college.
Yes, sir. Yes.
Mysore.
Sir, earlier I was working 12 years at Sirsi College of Horticulture, Sirsi. This is my student work. I'm chairman for the MSc student. That's why I'm presenting. So last year I completed that. So last year I got transferred to Mysore College.
Okay. Thank you. Any questions? No questions.
Thank you very much, sir, for kind opportunity.
Move to next presentation. Geetanjali. Geetanjali. She's here. Okay, please come. Serial number 19. 19. Geetanjali PA.
A very good afternoon to everyone present here. Myself, Dr. Geetanjali, Assistant Professor, Department of Microbiology, Field Marshal, Karepa College, Madikeri. The topic what I'm going to present today is the coffee has degradation potential of a litter harboring fungi, Mucus acinoloides GL1, through solid state fermentation technique. We all know that coffee is one of the popular beverages worldwide, and the production generates a lot of waste. And this is estimated to be about 10 million tons of coffee husk waste every year. The application of this husk is extremely restricted as it is a bulky product and it is rich in lignin, tannins, and caffeines. The polyphenolic compounds which are not degraded naturally in the environment. The improper disposal of this coffee husk waste also presents environmental risks and challenges to human health. Despite the promising potential of this coffee husk, little work has been done with microorganisms which can degrade them. The lignocellulosic microorganisms, which are found abundantly in soil, can be used to degrade and recycle these agro-residues, which are rich in lignocellulosic compounds. The composted material enhances the soil fertility and improves the soil health. Under this background, the present study was undertaken to study the efficiency of one litter hovering fungi which we isolated, the Mucus acinoloides species, to study its efficiency over the period of 60 days of degradation time. These are the objectives. We isolated this from the litter samples of coffee plantation itself, and the substrate coffee husk was also procured from a coffee a processing unit, and we adjusted the moisture percentage to 70%, and then under restricted condition we studied the various degradation potential of this particular fungi. So the results showed that the substrates were harvested during the 60 days of incubation period and studied for the various parameters like the pH, total organic matter, total organic carbon, nitrogen, crude protein, and then the phosphorus and potassium content. And we observed that there was a considerable decrease in the pH and decrease in the organic matter content and organic carbon content in the during degradation time, and there was an increased 48% of nitrogen content and phosphorus and potassium in the coffee husk. Then when we found out the C's to N ratio and then the C's to P ratio and C's to K ratio of the to know the degradation potential, we found that there was a considerable reduction in the C to N, CP and then CK ratio. There was a considerable decrease in the percentage of lignin content of the inoculated substrates when compared to the control. And also in this work, we studied the production of certain lignolytic enzymes, specifically manganese peroxidase, lignin peroxidase, and lactase enzyme during the degradation. These enzymes become active. They are very important for the degradation of these lignin-rich compounds. There was considerable increase in the production of these enzymes, which are having a tremendous commercial, the industrial applications. So this is the untreated coffee husk and then the treated coffee husk by the mucus species. So I would like to conclude that the enormous amount of agro waste generated in the agriculture fields could be converted into valuable compost. To accelerate the degradation rate of these lignin-rich agriculture waste, inoculation of lignolytic microorganisms is essential. And the native indigenous fungal isolates are better compared to the standard cultures or in the normal conditions for the local agro waste. And the widely available agriculture waste through solid state fermentation can be used as a potential for a production of potential lignolytic enzymes which have industrial applications. Thank you.
Thank you, Geetanjali. Any questions from audience side? So basically, how you fermented the product?
Yes, Koji fermentation.
Huh?
Yes, coffee husk was collected and then sterilized. Then we adjusted the moisture percentage to 70%. Actually, I've done a lot of study, different moisture parameters condition we have. And then incubated, sir, in that.
The causative agent for fermentation, what is the agent?
I have inoculated mucus acidoides. Yeah.
Because usually in other cases they use organic acids also for fermentation.
Yeah.
That becomes expensive, especially in the food grade items. You can use organic acids.
Enormous amount of coffee has to be this is the big concern for us if we don't know what to do with it. So as a compost, we can collect that and then inoculate this with the isolated organisms. Degradation is faster and otherwise this coffee has not much.
So that end product,
End product is a husk with less lignin content. When it is less in lignin content, naturally rich in phosphorus, potassium, and nitrogen, you can use it as a compost as well. Yeah, problem.
after fermentation, it can be used as a fertilizer.
The biggest concern of coffee is because of the high heat content. We can't decompose them, we can't throw them out. So it's a huge bulky product. We don't know. The present the farmers they throw it in their bodies.
The most they can use is a compost. Compost, no?
Even then it may be this. Then it can be this.
Thank you. Okay.
Yeah. You're working on it. I work in that way. I think work for studies.
21, right? Giving this one. Girimaleshwar, right? That was the Girimaleshwar Magari. Magari, right. Then one more last presentation. What is the name? Okay.
Good afternoon to everyone. So myself, Dr. Girimaleshwar Bagri, Assistant Professor, DRM Science College, Davangere. My topic is on study on diversity of spiders, Davangere city. So basically, spiders are defined as air-breathing earth arthropods with two body parts, cephalothorax and the abdomen. They belongs to the class Arachnida of the phylum Arthropoda that possesses jointed appendages and chitinous exoskeleton. Cephalothorax having the four pairs of segmented legs attached to the abdomen, unlike insects, Arachnida do not have the antenna. The order Arania, which includes all the spiders, is divided into two suborders. The one is Mesotheria and Aphisotheria. The order Arania ranks seventh in overall species diversity among all the orders of organism. Currently, more than 52,750 non-valid spider species are there. These species are organized into 136 families and 4,376 genera as per the late May this year, May 2025. But likely not exhaustive portion of the estimated 1,20,000 to 12 lakh total species thought to be exist. The significance of the spider is spiders play a significant role in both ecosystem and cultural narratives. They are beneficial predators controlling insect populations, and their unique webs inspire various aspects of human culture and technology. In addition, they hold a symbolic meaning in cultures, often representing fate, creation, or even good luck. Threats of the spiders. So, spider faces various threats, including deforestation, habitat loss, and climate change, use of extensive pesticides and fungicides, global warming, and competition from other animals. Some spiders also face the threat of being preyed upon by birds, reptiles, mammals, and other invertebrate species. So that we have to conserve the spiders because only the conserve focuses on protecting spider populations and their habitat due to their crucial role in ecosystem. Their potential for biological control, unique silk and venom properties. Key conservation strategies include habitat protection, land management, and raising public awareness about spider importance and threat they face. The main aim of this project is to present work was undertaken with the following objective. The chief aim of the work was to study about the spider diversity, the surrounding areas like DRM Science College and also as the Vishweshwara Park of Davangere district. The capturing of photos and identification of spiders from the mentioned study areas. The identified spiders are divided based on their families and distribution. So materials and collection was done by through the study was done between the month of April and June 2025. The spiders on the building walls, botany labs, rocks, and also a rural part of the garden crops like hibiscus, lemon, mango, and grasses, coli, basil, etc. The methods of collection, we spend a lot of time in the field observing in the natural habitat. We use the Sony DSLR camera to capture the images of spider without any harming disturbing them. We carried out notebook and pen to note the observation. We avoided some sudden movements that might scare spider away. We kept a safe distance and did not disturb them web or young ones. These are the methods we followed to capture images of the spider when we visited different places. So these are the 9 families we have identified. So the present study was conducted identification distribution of spider, Vishweshwar and as well as Davangere DRM Science College campus. The family Salatidae comprises larger number of species, means here 50 species we have identified, followed by the family Aranidae, Spiricidae, the families Lycosidae and Jorospidae, the families Cytosidae and Polycidae, and the families Dolomine, Pilodormidae. So the diversity of community has been studied in the different parts of the world. The present study may be the first attempt of spider fauna in the same area. The total of 43 species belongs to the 13 species and highest number of species in selected. The latest number of families are Dolomedidia and Philodormidia. The families namely Phyllocydia, Cytosidia, Aranidia, Leucocidia, Jorosidia, Sporocydia consists of a moderate number of species. The proper protecting measures should be taken in attention in order to minimize the natural habitat loss as a spider fauna independent upon accurate environmental conditions. And also, we related analysis should be conducted on a large scale to fully evaluate and explore spider fauna of same reasons as well as other hill station of district. So these are the references we follow. Thank you.
Yeah, thank you. So any questions? Yeah, so there are no questions. Thank you once again. The last presentation by Mr. Somalingappa. Somalingappa is here? Yeah, please come. So this is the last presentation, whereas other few presentations are there. I can just, yeah. So I will mention the names, okay? 31, Dinesh Upat. Then we have 41, Vijesh M. Then 48, Dhanashree. They are there in the other. Yeah, okay. Then 58, okay, that's what I'm telling you. Rinda. All these presentations will be done in the other seminar session too. No? Okay. If anybody is here, so no need to wait. That's why I mentioned. This is last presentation.
Sir, good afternoon, everybody. Sir, I am Somlingappa, B. Pallu. My topic is investigation on major foliar diseases of BT cotton grown in northern dry zone of Karnataka. So these are the objectives of my research work. Actually, before to go to the objectives of the research, actually the cotton plant is a Gossypium species, is a natural crop of it is a crop of very uh, very interesting plant because it gives us the textile, the fiber which is used in the field of the textile industry. Cotton belongs to the family Malvaceae. So the word cotton, it is derived from Arabic word kutan, means what? Fancy fabric. The plant is a shrub and is native to the tropical and subtropical regions around the world. Cotton fiber is very soft, fluffy, stable fiber that grows in a ball or a protective capsule around the seeds of the cotton plant of the genus Gossypium. Then cotton is considered as the king of the fiber because of the most superior fiber and garment qualities that can be made into a yarn for a textile uses. Then, due to its importance in the agriculture as well as in the industrial economy, it is also known as white gold. Cotton is grown in the world in an area of about 321.26 lakh hectares, producing 113.32 million bales with a productivity of 766 kg per hectare. In our country also, I have mentioned here. Then I will go to the next slide. So here I have shown objectives of the research work. Major objective of the research is that survey for the major foliar diseases of the BT cotton in the cotton growing regions of the north dry zone of Karnataka state, and then after disease severity of the major foliar diseases in different BT cotton hybrids, epidemiological studies on major foliar diseases of the BT cotton, and lastly, screening of the BT cotton and non-BT cotton genotypes for major foliar diseases. So methodology includes survey for the various foliar diseases of the BT cotton. Then actually, survey is an in-depth inspection of the diseased condition or a disease of a plant or a crop as a whole. It is a method of gathering the information from a sample of the individual plants. This sample is usually just a fraction of the population. Whole crop is being studied. Then here, survey for measure the foliar disease of the BT cotton in cotton growing regions of the north dry zone of Karnataka. Here, to gather the complete data on the disease development, its distribution, severity, and to recognize the hotspot for genotypes, diagnostics in the disease resistance screening program, a survey of the disease is required. Plant disease survey involves fixed plot survey and a rapid roving survey. Plant disease survey aims at identifying the causal organism from the symptoms observed and by laboratory confirmation. So the fixed plot survey and rapid are also called roving survey involve in situ assessment of the disease in the standing crop of a farmer's field. Supporting methodology also includes recording the weather parameters of the respective areas to study their influence on the diseases. So the fixed plot survey, in this method of survey, 2 crop fields of about 1 acre in size are selected in 2 different villages, which comes under the area to be covered for surveying. In each of the selected crop fields, 5 microplots, each of the size of 1 square meter area, are selected or fixed. These microplots are laid down, one in each 4 quarters of the field and in the middle, and should be fixed about 10 meters away from the bounds. In fixed plot survey, disease is assessed from the fixed plots selecting in a field. The data are recorded regularly from sowing till harvest of the crop from the same fixed plot in a particular field. And then data collected in these surveys are used for developing the forecasting models. So this is about the rapid, also called roving survey. In this method of the survey, assessment of the disease severity is carried out from a randomly selected spot in a short period of time over a larger area. It provides the information on disease level, which helps in determining the and adapting appropriate control measures. So I have selected in our Karnataka state there are 10 agroclimatic zones have been recognized. I have selected for my research work. It is north dry zone of Karnataka. So this north dry zone of Karnataka includes Vijayapura district, Gadar district, Bagalkote district, and also some part of Ballari district. Then rapid roving survey helps us to get information regarding the Alternaria leaf blight disease, and then the grey mildew disease and the bacterial blight disease. So these are the major the foliar diseases. For identifying for these the study of these diseases, actually I have used here. This is the Karnataka map showing the north dry zone of agricultural zone. So this is the north agricultural zone. So these are some of the photos taken during a survey work. Actually, a large number of viral bacterial than fungal diseases, even nematodes infect the cotton plant. So out of them, grey mildew caused by Remolaria gossypi is one of the major foliar disease. Bacterial bite, also called angular leaf spot disease, caused by Xanthomonas campestris, PV malvacerium, is also bacterial disease. It is also one of the major bacterial disease, foliar disease of the cotton. Alternaria blight, caused by the fungus Alternaria macrospora, is also one of the major diseases. So these are the fungal diseases, out of which Alternaria leaf blight and then Remolaria grey mildew are the major one. So here I have identified, and then I have identified the disease severity of the various diseases as I have mentioned earlier. The cotton fields were randomly selected in a village on the survey route. In each field, actually the plants were selected at a random, and the severity of the disease were recorded. The percent disease incidence was assessed by counting the number of the plants showing Alternaria blight, grey mildew, and then the bacterial blight. By using this formula, percent disease index incidence, it is also is equal to number of the plants affected into 100 divided by total number of plants observed. And then severity of the disease was estimated based on the disease severity scale 0 to 4 given by the CRR in the year 1988. Then this is about the alternaria leaf spot disease. So this is the chart or the table. Here in this table I have listed, I have shown the PDI percentage disease index of the various diseases, and then causal organisms and their morphology and symptoms of the various the fungal disease, ultra-rare blight, and then grey mildew, and as well as bacterial blight disease. So a rapid survey was conducted in 5 districts of northern dry zone of Karnataka, encompassing Gadag, Bagalkoti, Vijayapura, Koppala, and then Ballari. Prevailing diseases were recorded in different farmers' field at the 3 different stages of the crop growth, namely seedling, ball initiation, and then ball maturation, by using the scale as described earlier in the materials and methods. In Gadag district, the maximum severity of the alternaria leaf blight, it is 20.35 PDI percent disease index. Grey mildew, it is about 15.06 PDI, and then bacterial blight, it is about 16.50 PDI, and were recorded in the ball maturity stage on the cotton plant. In the similar way, in Bagalkoti district, Vijayapura district, Koppala district, and then however in the Ballari district also, the PDI result I have shown here in this the result. And lastly, I would like to conclude my the presentation here. Cotton is known to be infected by many foliar diseases. The major diseases that are posing a threat in the production are alternaria leaf blight disease, grey mildew, and the bacterial blight. As these major foliar diseases are taking upper hands in recent years because of the increased severity in the northern dry zone of Karnataka, the present investigation included the survey for the major foliar fungal diseases and bacterial diseases of the cotton, epidemiological factors that are affecting the disease development, and screening of the cotton genotypes against the diseases affecting the cotton plant, where the highest incidence of the fungal diseases were recorded in Koppala district. It was also observed that bacterial blight incidence recorded least recorded in Vijayapura district. So these are some references. Thank you very much, sir.
Hey, thank you, Somalingappa, for your nice presentation. Can you go back to your title page? Title page. Yeah, this is your Kuala University, right?
Yes, sir.
So your Department of Botany and Seed Technology.
Yes.
In Sayadri College. Sayadri College is an independent college, right? Is it under the university only? Kuala
University Constituent College, sir.
Constituent
Kuala
College.
University Constituent College.
Affiliated college or constituent college?
Constituent College.
Oh, okay.
Kuala University, once it was started earlier,
Right.
Sayadri Science College, sir.
Okay.
So I am doing my research work under Dr. H.N. Ramesh Babu, sir.
Okay, thank you.
Thank you, sir.
Thank you.
Thank you very much.
So that completes all the presentations that are listed here. Since yesterday and today, 2 day presentations in various technical sessions has been completed. And I think we have also have to evaluate for best paper presentation and all. So that so all the chairman and co-chairman, they will give their evaluation sheets. And probably I think we have to meet here again at 4:30 for the validatory function. So we have some time. You can some tea break. You can have the tea and assemble back here. Okay. On behalf of Madam Nalini is there, so I thank her for her support. This session. Thank you for your all nice presentations and patient hearing and your involvement in discussion. And, okay. Thank you all.